Could I have your attention, please? Can everyone hear me okay? Yes. Yeah? All right. So, welcome to NEURON. So, it's our 29th NEURON Conference. Here we're hosting it at Quinnipiac University, and so I wanted to extend a warm welcome to all of you. Just so you know where you are, you're at Quinnipiac University. It's about 6,500 students or so, and we have a new medical school, which is where we're currently having our talk and all of our workshops. The conference itself is primarily funded by the College of Arts and Sciences. So Dean Smart, you may see him wandering around later this afternoon. They're helping to foot the bill in addition to your registration fee, so thank you all. In terms of who's actually running it, so Dr. Bulger, she's going to introduce our speaker today, and she's the chair of the psychology department. I am the local organizer and the director of the behavioral neuroscience program this year. And then Dr. Betts, who's run it for a number of years, and she was helping run it. And she has her daughter here, Maeve, with her, so that's pretty awesome. If you want to find out more information, again, I'll send you to the website, which you should be able to find. And then I also want to point out that past speakers have had their talks recorded, and you can find them there if you're interested. So last year, we had Dr. Sinha, and she gave a great talk. In terms of registration, here you all are, so I wanted to extend a thank you. We're a little last minute, but it's good, you're all here. And then in terms of geographic representation, we have representation from all over the Northeast. So I appreciate all of you that took the time to make the long drive this morning. We're hoping it's going to be a great conference. And finally, I would like to thank all of the workshop presenters. They're going to be presenting 4 different workshops after this, and you can find the room numbers here in your program. There's also a map. So the map, this is basically where you came in, and you'll be going up to the 2nd floor in order to get to most of the workshops. If you're staying for the career panel workshop, that's going to be in here, okay? So with that, I'm going to hand this over to Dr. Bulger to introduce our speaker.
Thank you. Let me add my welcome to all of you and thank you for coming. We are delighted to host again this year and to see so many people here this morning. It's fantastic. I'm going to keep this short and sweet because you have a very nice biography of Dr. Addy in your program. So please allow me to just give a few highlights of you. Dr. Addy is an assistant professor of psychiatry and of cellular and molecular physiology at Yale University. Apparently he likes to be busy. After earning his bachelor's degree from Duke and a PhD from Yale, he held a postdoctoral fellowship at the University of North Carolina, Chapel Hill, and he's now back at Yale as a faculty member, lucky for us, because he could come here today. Today we're going to hear about some of the research coming out of the Addy lab at Yale, and they have a very nice website that helped me out a lot last night when I was getting ready to come talk to you about you. The Addy lab focuses on understanding the neurobiological underpinnings of learning and control, especially motivational control, particularly as these are changed in psychiatric conditions, and that's some of what we're going to hear about today. Those psychiatric conditions that his lab seems most interested in include depressive disorder and substance abuse. Dr. Addy is widely published in many academic journals that are important in neuroscience, including behavioral brain research, psychopharmacology, and neuroscience. He's also a member of several professional societies, including the Society for Neuroscience and the American College of Neuropsychopharmacology. I just wanted to name that one because that's a really hard word to say. Dr. Addy engages in scientific outreach in many areas, which is something we really appreciate here at Quinnipiac since we are so focused on undergraduate education. We take a lot of time to engage in that sort of outreach, and in the psych department we call it giving psychology away. You're giving neuroscience away. We really appreciate that. So he does presentations at career days around the local area and takes part in brain awareness events, and he has also mentored some local high school students. So we're going down to that level of outreach as well. We're very honored to have you here today to share your work with us, and thank you very much.
I have one last announcement for the faculty. So if you're interested in judging during the poster sessions, you can find Denise McQuaid at 1 o'clock outside around near where the posters are. So I encourage you to do that.
Excellent. Is that on? I think it is. Can you guys hear me all right? Excellent. Well, thanks for having me here this morning. I'm really impressed to see such a large crowd early on a Sunday morning, so I think that speaks well to you all in terms of your enthusiasm. I've actually been involved in Neuron one time in the past. In 2010, I gave one of the mentorship workshops, so it's great to be back as a keynote speaker. Really honored to be here to talk with you this morning, and it's been fun to actually interact with some of you already, even in the brief time that I've been here. So as they've mentioned, Dr. Bulger and Dr. Ahern, I'm going to be focusing today, telling you on some of the work in my lab, looking at the neurobiological mechanisms of substance abuse. And in particular, we're focusing on trying to understand how cholinergic and dopaminergic mechanisms are important for drug craving and drug relapse. Now, one thing that I've noticed over the years is that whenever I tell people what I do for a living, it actually seems to be a good conversation starter. That might be a little bit surprising to you. Science isn't necessarily something that people are talking about on a daily basis. But I think part of that is because of the topic of what we study. So in particular, looking at substance abuse. And most of the people I talk to either know someone, a family friend or a relative who struggled with substance abuse at some point in their lives, or they struggle with it themselves. So in that sense, I think this is a really relevant topic that a lot of people are still thinking about. People really want to try and understand what happens during substance abuse, what are ways to try and prevent that substance abuse, and importantly, to try to treat it once it's occurred. So that's what I'm going to be focusing on today as we go through the talk. I also wanted to say, I know we'll have some question and answer time afterwards, but please feel free to interrupt with questions as we go through, and I'll also be looking for feedback from you all as we go through the talk as well. So before I get into the specifics of what we study in the lab, I just want to mention that in the broad sense, what my lab is interested in understanding is the neurobiological mechanisms of complex behavior. And as it's already been alluded to, we're particularly interested in behaviors and maladaptive behaviors that are associated with psychiatric illnesses. So the two main areas that we focus on are depression and substance abuse. Obviously, for today's talk, I'm going to be focusing on our substance abuse piece. But a lot of the basic biology that I'm going to be talking about seems to be important for both of these types of disease states. And so that's something I want you all to keep in mind as we go through the talk as well. So to put this in a little bit of context, I also wanted to show some brief statistics, both in terms of drug use and drug abuse in the United States in particular. So this is from a survey that was performed by the Substance Abuse and Mental Health Services Administration in 2013. This is surveying individuals 12 years of age and older. And what they're showing you here, whoops, wrong button. What they're showing you here, just making sure everyone's paying attention, is the amount of drug use within the past month. So again, individuals 12 years of age or older, and you can see that if you just categorize illicit drugs in general, it's around 25 million users within any particular month. You can, of course, break this down into categories: marijuana, psychotherapeutics, cocaine, heroin. And you can see these numbers range from the low millions to about 20 million or so. I should also mention this doesn't include smoking, so that ranges is upwards of about 40 million users. So you can see it's still a very prevalent problem in this country. Now, of course, this is talking about drug use. What we're particularly interested in is drug abuse, and we'll define that as we go through the talk. But if you look at those numbers in terms of how many have used in the past year, you can again see that these numbers range from the high hundreds of thousands of users to millions of users in any particular year, any particular point in time. So this has a lot of different consequences that's associated with it, obviously. One of them is the fact that this drug use and drug abuse is also associated with increased risk for other types of diseases. So in particular for cardiovascular disease, for various neurological diseases, and in cases of overdose, you can actually have death as well, which obviously can be detrimental on several levels, especially to family members and friends. Another aspect that we often talk about is the fact that those who abuse these substances often have higher rates of and a higher instance of violence and unemployment. So obviously this has detrimental societal, socioeconomic, and family consequences as well. And for today's talk, as I mentioned earlier, I'm going to be focusing particularly on drug relapse. And the reason for that is that this drug relapse itself turns out to be one of the main obstacles to successfully treating these individuals. So you have folks that are oftentimes motivated to try and get off a drug, but end up relapsing, and obviously that continues this cycle and continues them in this addictive spiral. Now this relapse can actually be initiated or precipitated by different factors. I just want to mention some of those briefly. For most of today's talk, I'll be focusing on drug-associated cues. So cues that can actually induce drug craving and drug relapse in these individuals. But you can also have other things that can precipitate relapse. So one of those is stressful situations. So for instance, let's say you have a smoker who has a stressful situation at work, or someone who has a stressful final exam coming up. All those things can be enough to lead someone to relapse into using the drug again. And the last aspect I want to also highlight is re-exposure to the drug itself. So this is an instance of someone who has tried to abstain for a certain period of time, decides, oh, I'm just going to take that one more hit. That one hit turns into a second hit, which turns into a third hit, which turns into a fourth hit, and before you know it, that individual has relapsed as well. So just want you all to keep these different aspects in mind as we go through the talk. But again, for today, I'll be focusing primarily on this aspect of drug-associated cues and how that's important both for craving and the eventual relapse as well. I'm sure that you all have had exposure to this in your biology classes and neuroscience classes so far, but when we talk about addiction, it's similar to what we see with other diseases in the sense that the interaction between the environment and the biology is critical to actually developing the disorder. So in terms of, again, as I mentioned, the environmental factors with addiction are particularly important. And I wanted to just, before I go through and give you some examples, just wanted to get some ideas from the audience about what factors or characteristics you think in the environment could actually enhance someone's susceptibility to developing addiction.
Peer pressure.
Peer pressure, yep, that's a good one. Another example. Stress, yep, definitely a good one. All the way in the back? Economic status, yeah, so there is differences in terms of susceptibility across economic status as well. Another idea?
Abuse in the family.
Mm-hmm. So family history, great.
Accessibility to the drugs?
Exactly, yeah, accessibility is also another important one. How about a couple more?
Violence.
Violence, yep. So can you expound on that a little bit?
Perhaps, it could be sexual violence or it could just be violence in their environment if they're growing up in an environment that, say, has higher gun crime rates.
Exactly. All those aspects, again, can influence susceptibility and is very tied to stress as well, which someone else mentioned. Great example. One more?
Chronic medical concerns, medical conditions.
So expand on that a little bit as well.
Somebody who has chronic pain would probably be more likely to use certain drugs, or some people who have chronic conditions and are always in the hospital are more likely to use certain drugs.
Right. That's a great example. So you guys have hit on a lot of key points which I was going to bring up in the slide, so thanks for helping me in advance. So some of them that you've already mentioned, so I think someone actually mentioned social interactions, someone mentioned stress as well. We also alluded to the cues earlier. So you're all on point for an early Sunday morning. So of course, as I already alluded to, biology is also a very key component. So you've hit on some of these as well already, but any other examples of what you think might be increasing susceptibility in terms of biology?
Genetic predisposition.
That's a great example. Genetic predisposition was the example.
Personality, how maybe risk-taking behavior.
That's a great example as well, risk-taking behavior.
Genetic modification.
Expand a little bit on what you mean by that.
So environmental factors such as stress can trigger changes in genomic organization that can potentially shut down or overstimulate certain genetic pathways that reorganize the neurobiology and neurochemistry in such a way that addiction becomes harder.
Great example. Well said. Do you want to come up and do the rest of the talk? One more example?
Situations.
Give me a little bit more in terms of a particular type that might fit there.
Someone being backed into a corner without knowing how to get out, so they might resort to violence.
Yep, it's a great example as well. So I might need not even to do the rest of this slide, but I will just in case. So you guys did mention genetic factors. That was mentioned. That's a great example. Drug sensitivity, which I think someone was alluding to as well. So the first time that someone is exposed to a particular substance, someone might find that rewarding, someone might get high off of that, but another individual might actually find that aversive, depending on their particular drug sensitivity. Differences in circadian rhythms can also contribute to someone's susceptibility to addiction. Disease states, and I think people have alluded to that already. So there are certain diseases that are known to be comorbid with substance abuse. And sex as a biological factor is also very important. So male versus female, that can lead to differences in susceptibility as well. So obviously the biology interacts with brain mechanisms and circuits, which can, again, influence someone's susceptibility to addiction. But of course, one obvious and key aspect here is exposure to the drug itself. So different from some of the other diseases that we often talk about in biology, when we're talking about substance abuse, the user is actually an active participant in getting the disorder, in the sense that you have to be exposed to the drug itself before you can develop drug addiction. And then, of course, we've also alluded to history. I think there have been a couple examples that people have mentioned in terms of how previous personal history or family history can actually influence someone's susceptibility to addiction. Also, these aspects of history can influence someone's expectation about what happens or what's beneficial or detrimental about using a particular substance. Learning is also another critical component. We'll talk about that more as we go through the talk. But again, all of these different aspects can actually impinge upon the brain mechanisms. And the key aspect that I wanted to also highlight here is that it's the interaction, of course, between the biology and the environment and the brain mechanisms that can all lead to the addiction. And the addiction itself can also influence these other aspects. So someone's addictive disorder can influence how they act or interact in the environment, and can also affect their neurobiology, which then feeds back and affects and can sometimes make the addiction that much worse. So just something to keep in mind as an underlying theme as we go through the talk this morning. So I also wanted to just go through some of the symptoms that are typically associated with substance use disorder from a diagnostic standpoint. And so historically in the field, most of this has been based on the Diagnostic and Statistical Manual of Mental Disorders. This is the 5th iteration of this document. There has also been a push in the field recently to try and move away from just using this straight symptomology, but also to try and think about the underlying domains that are associated with substance abuse and with several other types of mental health disorders, and trying to understand the biology that's contributing to those differences. So really trying to get a better sense of the biology that's important for mental disorders in particular, without a focus on symptoms per se. But I thought for today's talk, it would still be good for us to try and talk about some of the characteristics that are important in terms of symptomology. So before I have you all give me some examples of characteristics that you might be thinking of, just wanted to give you the brief definition that's included in the DSM-5. So in terms of substance use disorder, DSM describes it as a maladaptive pattern of substance use leading to clinically significant impairment or distress as manifested by 2 or more of the following symptoms. And so there's a scale that's used. So if you have 2 to 3 of these symptoms, it's considered mild substance use disorder. If you have a few more of the symptoms, it's moderate. And if you have even more, then it's severe. But before I go through and show you the list of what I have here, any ideas or thoughts about specific characteristics or situations or circumstances that you would associate with symptomology for substance use disorder?
Withdrawal headaches.
Expand a little bit on that.
So a person who becomes more dependent on this drug to maintain a certain function in the brain when they are without the drug, it puts them under stress and they develop a headache.
Yep, that's a great example. Any other thoughts? All the way in the back?
Continuing use despite negative social, family, economic consequences.
Yep, great example.
Tolerance.
It's another great example as well. Any other ideas?
Stereotypical behavior.
Can you describe that in a little bit more detail as well?
Like maybe with amphetamine use, you'd have like continuous repetitive like pacing or other stuff like that.
Yep, great example. Right here.
Using substance when someone's alone.
Say it one more time.
Using substance when someone's alone.
Yep, that's another thing that happens fairly frequently. One more example.
Denial.
Denial? Great, so you guys again have hit on several of the key points. So just to go through, so there's actually 11 different characteristics that are listed in the DSM-5, just to go through those briefly, and some of them you've already mentioned. But one, again, is this recurrent substance use that results in failure to fulfill other major obligations at work, school, or home. So there's a shift in the behavior towards the substance use as opposed to normal obligations that are important. Another is recurrent substance use in situations where it's actually physically hazardous to use that substance. A third, and I think we've alluded to this a little bit, is continued use of a substance despite the fact that there may be persistent or recurring social or interpersonal problems that are caused or exacerbated by that substance use. Tolerance and withdrawal are 2 aspects that you all mentioned as well. So tolerance in the sense that someone might feel the need to take more and more of the drug in order to achieve the same high they initially got with a lower dose of the drug. And also, if they do use the same dose of the drug as they did initially, you actually see a decreased or diminished effect of that continued use. Withdrawal, on the other hand, is what typically happens when people have abstained for the drug for a period of time. So this can be either physical symptoms or psychological symptoms that are associated with removing the substance for a particular point of time. And in some instances, you can also see that if someone takes a closely related substance, that can actually relieve some of these withdrawal symptoms. So another key aspect as well. Another piece, which is somewhat related to this idea of tolerance, is that oftentimes people will take larger and larger amounts of the substance over periods of time. often taking larger amounts than they initially intended to. So someone who goes into a specific situation, wants to take a certain amount of the drug and ends up binging, for instance. And oftentimes there's also a persistent desire for someone to quit. So you see this a lot of times with smokers who might say, oh, I really want to quit, I'm trying to quit. But despite their intentions or efforts, there's a major challenge to actually cutting down or controlling that use. Another piece which I think you all have alluded to is the fact that there's often a great deal of time that people will spend in activities that are necessary to actually obtain or use the substance or to recover from its effects. So in that sense, it becomes very life-consuming. Oftentimes, people will, again, start to avoid important social, occupational, or recreational activities, giving up those activities and replacing them with activities that are important for their substance use to continue. A 10th aspect is the fact that there's often continued use, and I think someone alluded to this, despite the fact that there is knowledge that there are physical or psychological problems associated with that use. So you might talk to someone, they might say that they know that this is causing lots of problems in their family life, in their personal health, but they just can't break the habit for whatever reason. And again, the piece that we'll focus on for today's talk is this idea of craving, so having a strong desire or urge to use a specific substance. And in talking to individuals who've been struggling with this at certain points in time, they'll say it's like wanting to have your favorite food, but at a much higher level and not being able to have that food, just that intense craving that they have for the substance itself. And I've mentioned a few of these points earlier, but again, just want to reiterate that when we're talking about substance abuse, it's always important to keep in mind that the individual is an active participant in getting the disorder in the sense they have to use the drug in order to become addicted to it. And another key aspect is that this is only a subset of individuals who are using drugs. So if you remember the first slide that I showed both drug use and drug abuse in this country. So not everyone who uses a substance will transition into an addictive state. The most clear example of that is with alcohol, for instance. So in the US, 50% of those individuals 12 years or older, which is a very low age from my standpoint. I was surprised to see that. But regardless, 50% of individuals 12 years or older use alcohol, but only 7.7% actually will develop alcoholism. So clearly there's a subset of people that will develop into this stage. We talked a little bit about susceptibility early on, but it's something that's always important to keep in mind as we go through. And there's a lot of work trying to understand what are the factors that can enhance susceptibility, both from social and environmental standpoint, but also from a biological standpoint. So are there differences in biology that can increase or decrease someone's susceptibility for developing a substance use disorder? So now I want to transition and get into a little bit more of the underlying neurobiology and then start to talk about some of the work that we've done as well. And this probably isn't a big surprise to you, but dopamine has been a key focus of a lot of this work over the years. And so if you go to the National Institute on Drug Abuse website and just look at what they've presented, this is, again, information they're trying to present to the general public so that people can try to have a better sense of what's contributing to substance abuse. You'll see a figure like this. So here what they're highlighting is the fact that dopamine is important and critical for brain reward pathways. So what they're showing here is just a mid-sagittal slice, so through the middle of the brain here, and showing this dopamine system. So this originates with dopamine cell bodies, which are located here in an area of the brainstem as the ventral tegmental area. They send their neurons to areas which include the nucleus accumbens and the frontal cortex. Now, as many of you may already know, dopamine neurons are also important for movement. So there are dopamine neurons in the neighboring substantia nigra, which project to the dorsal striatum. And these are neurons which are compromised in things like Parkinson's disease and Huntington's disease. Here they're focusing on a different subset of neurons, and again, their importance in terms of reward. So what they're showing you here on the right is the fact that drugs of abuse can actually alter a dopamine signal in the brain. And obviously, this system wasn't set up to detect responses to drugs of abuse. It was set up for natural rewards. So if you have an instance where a human or an animal even is consuming food, what you see is that there's increased dopamine that's released in the synapse, so between the presynaptic and postsynaptic cleft. That dopamine binds to receptors, and you have a hedonic or pleasurable experience that's associated with food. I'm sure if I asked you for examples, you all could give me lots of different examples of your favorite food, and I would bet that dopamine is probably involved in your response to those foods. So what we often talk about in terms of the drug abuse literature is that drugs of abuse hijack natural systems in the brain. So what they're showing you here is an instance that you see with cocaine. So now you have cocaine that's present in the body. It can actually block these dopamine transporters, prevents dopamine from being removed from the synaptic cleft, And now you have a much higher level of dopamine that's available in the synapse compared to what you see with a natural reward like food. Now, if you think back to the symptoms I just talked about on the slide previously, one thing that I mentioned is that oftentimes individuals will engage in using substances in place of their other natural rewards. And you can see from a physiological standpoint that most likely the pleasure or hedonic valence that's associated with this cocaine is going to be much higher than what you would have for a natural reward like food. So again, the key point that I want to reiterate here is the fact that these drugs of abuse are actually chemical ligands that interact with neurotransmitter systems in the brain, which changes the way people feel by altering this neuronal signaling. So again, this is highlighted here. This is work from a study that was done using microdialysis in rats, and basically they're just measuring dopamine levels in the nucleus accumbens after exposure to either food or drugs of abuse. So you can see that point pretty clearly. That you have a modest increase in dopamine that occurs when the animal is feeding for 20 minutes. If you inject the animal either with cocaine or with amphetamine, you see a much higher increase in dopamine levels. So a supra-physiological increase in dopamine, which again is associated with a high or a hedonic response to that drug of abuse. So there's a lot of information on this slide. I don't want you guys to try and take in all of it, but the main point that I'm trying to make here is that all drugs of abuse interact with this system and enhance dopamine signaling in some shape or form. So again, showing the ventral tegmental area here and the nucleus accumbens, all these drugs of abuse increase dopamine, but they all do it by different mechanisms. So in the case of cocaine, as we talked about earlier, it actually can block the reuptake of dopamine, allowing for more dopamine to be available in the synaptic cleft. In the case of methamphetamine and amphetamine, you can actually change the dopamine transporter in the sense that it's no longer taking up the dopamine, but actually releasing dopamine. So another way of increasing dopamine activity in the system. For things like nicotine, ethanol, THC, which is the active ingredient in marijuana, and morphine, they have various ways of actually stimulating these dopamine neurons, again, leading to an enhancement of dopamine that's available in the synaptic cleft. So again, all these different components can actually increase the dopamine that's available, and that contributes to the high that's associated with these drugs of abuse. So now that I've given you a little bit of the background in terms of the rewarding properties of the drug, I wanted to shift to what we've done looking at this idea of the neurobiology of craving. And one thing that's been noticed over the years is that dopamine is also important for these aspects of craving as well, not just for the rewarding properties of the drugs. So I have a few pictures that I'm showing you here, and the basic idea is that for a primary reward, like a drug of abuse, you have an unconditioned stimulus that's repeatedly paired with a conditioned stimulus. So in the sense of this animal here in the wild, this animal has probably learned over time there's certain discrete or environmental cues in this certain area that are associated with availability of food. For several of you in the room, perhaps you're having a coffee craving at this moment in time. So in the general sense, this is really just a machine of plastic and metal, but for some of you, this is a very salient cue in the sense that it tells you that coffee is available. And hopefully, the other thing that happens whenever I give this talk is that people start to self-evaluate and try to figure out where they have a caffeine addiction. I'll leave that for you all to decide as we go through. Obviously, for individuals struggling with substance abuse, you have drug paraphernalia or certain contacts or friends that now have become associated with the drug. So again, this idea that the drug is the unconditioned stimulus that's paired with all these other cues. And so in the sense, after that pairing occurs, these conditioned stimuli can now start to elicit craving and promote reward seeking in and of themselves. And one point that I really want to drive home here is that this ability of conditioned stimuli to drive this is true both for natural rewards and for drugs of abuse. Another aspect that people have started to focus on over the years is the fact that these conditioned stimuli, in some instances, can actually become desired or wanted in and of themselves. So one example I usually like to give is that of a smoker. So if you talk to a smoker who's trying to abstain for a period of time, we'll talk about how they miss having that hit every time they smoke a cigarette. So that hit of the hot sensation, burning sensation that they get at the back of their throat. Now this hit isn't actually something that's directly related to the reinforcing effects of nicotine, but the fact that these individuals start to desire or start to miss that feeling shows you again that this conditioned stimulus that's associated with nicotine actually can become desired or wanted in and of itself. And you may have noticed that there are a lot of e-cigarettes that are starting to hit the market recently, and that's one of the complaints that some people have had, that they don't have the same hit, the feeling that they have with a traditional cigarette with an e-cigarette. So again, these conditioned stimuli can become very important, both in terms of the craving and in terms of drug-taking behavior itself. So finally, getting to our research within this area, two of the things that we've been focusing on recently have been to try and identify the underlying neural pathways and neurobiological mechanisms that actually mediate this cue-induced drug craving and relapse. And not only to identify those, but to try and identify potential novel therapeutic targets that can be used to limit or prevent this cue-induced drug craving and relapse. So again, going back to this whole idea of dopamine being important not only for the rewards itself, but also for the cues, there were some seminal studies that were performed in the late 1990s, early 2000s that started to look at this in clinical populations. So basically what they're doing here is looking at cocaine-dependent individuals, doing brain scans, and looking at the activity in the brain, either when these individuals are shown a nature video or when they're shown a cocaine-associated video. And what they found is that during the cocaine-associated video, they actually saw increased activity in two areas in the brain, in the amygdala and an area of the prefrontal cortex known as the anterior cingulate. And both these are dopaminergic regions. So there have been lots of other studies that have replicated this finding, and basically what all these studies have found is that there is activity, increased activity, in dopamine regions such as the prefrontal cortex, striatum, and amygdala in response to the presentation of drug-associated cues. And this has been shown for several different drugs of abuse. So for cocaine, for alcohol, and for opiates. Again, highlighting the fact that this dopamine is important for some of these aspects. Now, what others have done is to also look in animal models to try and get a better sense not only of the brain regions involved, but the certain types of neuronal responses that are occurring within this dopamine system and how that may be important for some of these responses. So here, what I'm now actually showing is a mid-sagittal slice from a rat, again, highlighting the ventral tegmental area to nucleus accumbens and prefrontal cortex pathway. But what I want to do is just to briefly review the types of firing patterns that are observed in these neurons. So in the VTA in particular, there was work that was done in the late 1980s that showed that these neurons can fire in two different patterns. So one is known as this tonic firing pattern that happens at a rate of 3 to 7 hertz. So basically you have these neurons that fire 3 to 7 times per second at a pretty regular fashion. But at specific points in time, you actually see that these neurons will change their firing patterns. So they'll fire upwards of 15 to 20 times per second in irregular patterns, which are known as burst or phasic firing patterns. Now, people also want to know whether this had consequences in terms of the neurotransmitter release. So they also used techniques to record in the nucleus accumbens and see whether there were different patterns of dopamine release. What they found in doing that is that this tonic firing is associated with steady state basal dopamine release at steady state concentrations, whereas that burst or phasic firing results in rapid increases in dopamine concentrations that happen over short periods of time. So I'm showing the second time scale here. And so you can see that there's a very dramatic difference in terms of the type of response that you see both at the level of the ventral tegmental area and at the level of the nucleus accumbens. But of course, people want to know whether that had any important physiological consequences. So in these experiments, people actually looked at what types of firing properties and what types of neurotransmitter properties do you see in response to rewards and in response to conditioned stimuli that are associated with rewards. So this first set of experiments was actually performed in macaque monkeys where they're recording from the dopamine neurons in the midbrain while the monkeys engage in a particular task. Here what they're doing is to actually present juice flavoring as a rewarding stimulus to the monkey. And what I'm showing you here on this axis is basically all these marks indicate the firing of the cells. So on the top here, they're actually showing a summation of that firing activity. And this increase in activity here is an increase that's responding to, or it's an increase as a result of increased burst firing of these neurons. So when they provide this unexpected reward here, they actually see an increase in the burst firing in, again, the ventral tegmental area. Now, I've talked a lot about how conditioned stimuli or cues can be important. So what they did here, again, is to provide a conditioned stimulus that preceded the reward every time. So similar to what Pavlov did in his original experiments with the bell, signaling the availability of food. Here they have a conditioned stimulus that stimulates or indicates the availability of an expected reward. And when you do that, what you see is you now have an increase in the dopamine response in response to the conditioned stimulus itself, and not much of a change in response to the reward. Actually, if you omit an expected reward, you'll see a decrease in the neuronal firing. So again, highlighting the dynamic nature of this dopamine system to respond both to unexpected rewards and to cues that are associated with rewards. People have also looked at this in terms of the response to neurotransmitter release in the nucleus accumbens and see effects that mirror what you see with the cell firing. So again, a dramatic increase now in neurotransmitter release in response to a sucrose pellet in a rat model. If you do the same experiment, do a conditioned stimulus that precedes this reward, you again see a shift where you now have an increase in neurotransmitter release in response to the conditioned stimulus and not much of a change to the reward. So again, highlighting the dynamic nature of this system. So this is all showing correlations, but what we were particularly interested in my lab was to try and understand whether this phasic dopamine activity is actually critical for driving these behaviors. You see these responses to rewards, to reward predictive cues. Does it actually drive the reward behavioral response? And if it does, can we actually examine processes that regulate this phasic activity, and will those processes be sufficient to regulate cue-induced drug-seeking behavior? So those are the two main questions which I'm going to kind of take you through in terms of what we found so far. So if we're going to try and ask whether this phasic activity is important for cue-induced drug-seeking, we of course need a way to be able to look at cue-induced drug-seeking. So for that, we've turned to a rodent model. I know several of you are actually using these in your experiments as I looked through the posters, so that was nice to see as well. But for those who may not be familiar, basically what we're doing here is placing a rat in an operant chamber. The rat has two levers that are available in that chamber. One lever we'll term the active lever. Every time they press this lever, they're going to receive an intravenous infusion of cocaine. They're also going to hear a tone and have a light that's serving as a conditioned stimulus that's paired with that cocaine delivery. There's also an inactive lever. Every time they press that lever, there's no programmed consequence that occurs. So what we see is that if we place the animals in this paradigm, they learn very well and very quickly which lever is associated with cocaine and which lever is not. And as you can see over a 10-day period, they're very good at titrating their desired amount of cocaine. So you can see over these last few days, they're pressing about 20 times during this 2 hours to receive those cocaine infusions, and they're not pressing much on the inactive lever. And this model in itself has pretty good face validity in the sense that the rats will perform this behavior for drugs of abuse that humans will administer as well. So again, now we have a metric or a way that we can look at drug-taking behavior in these animal models. But we also want to look at drug-seeking behavior after periods of withdrawal. So for that, we perform an experiment that's known as cue-induced cocaine seeking after forced abstinence. So after the animals have learned the behavior here, we'll take them through various periods of withdrawal, anywhere from 3 to 45 days of withdrawal where they have no access to the drug and no access to the cues. And this is very similar to what would occur in a human who's going through detoxification, for instance. So after that withdrawal period, we'll then place the animals back in the chamber, again, allow them to press the levers, except this time, every time they press the active lever, they don't receive any cocaine. They only receive the conditioned stimulus that was paired with cocaine. Again, the inactive lever has no permanent consequence. And we use this as a way to try and see how much are they willing to continue to press this lever now just to receive the cue that was associated with cocaine. And we use that as a metric of their cue-induced cocaine seeking. And as you can see, this behavior is pretty robust over a long period of time. So anywhere from 1 withdrawal day out to 45 withdrawal days, you see they press the active lever much more than the inactive lever. So it's a very robust response that we see in these animals. So we have our behavioral setup. I also mentioned that we wanted to see whether this phasic dopamine activity is important for these responses, so we have to have a way to be able to stimulate and record this phasic dopamine activity. And for that, we turn to a technique known as fast cytometry, which is an electrochemical technique, and it basically just takes advantage of the fact that dopamine is electroactive, so it can be oxidized and reduced. Now, I'll try not to go into too much chemistry, but just to give you a little bit of an overview. So what we'll do is to actually take a glass microelectrode, and we have a carbon fiber that extends from this electrode where we actually measure our dopamine response. So this is 5 micrometers by 75 micrometers in length, so it gives us very high spatial resolution in terms of measuring these responses in the brain. And what we'll do is to actually apply a negative potential to this carbon fiber microelectrode, which attracts these positive dopamine cations. So we'll initially hold it at about minus 0.4 volts. When we attract the dopamine cations to the electrode surface, we'll then actually apply what's known as a potential waveform, where we'll ramp the potential from minus 0.4 up to 1.3 volts. And at a specific potential, dopamine will actually be oxidized to what's known as dopamine orthoquinone, and that will result in an electron transfer, and we can actually measure that transfer of electrons in terms of current. So basically what we're doing is just applying this potential, seeing how much dopamine is converted, and measuring the current that's associated with that. Once we get to this 1.3 volts current, or potential rather, we'll then ramp back down to this minus 0.4 volts, And on that sweep, this dopamine orthoquinone will be reduced back to dopamine, will then again have an electron transfer associated with it. So we're not changing the dopamine in the system for a very long period of time, but it gives us a way still to be able to measure the amount of current. And the amount of current that we see is directly proportional to the concentration of dopamine that's in the brain. So basically, when we're applying this, we apply it at a very fast rate, so we can actually apply this waveform 10 times per second. So basically, every second, we have 10 different snapshots of the dopamine concentration in the brain, and in that sense, we get a pseudo-real-time picture of these dopamine responses. Is everybody clear on that so far? So I also wanted to show you how we depict the data. So here, what I'm showing now is potential in this plane and the current in this plane. So as we go from minus 0.4 volts to 1.3 volts here, we see a dramatic increase in the current at about 0.6 volts. This is the point where dopamine is actually oxidized. So you can see that oxidation results in this increase in current. As we go from 1.3 volts back down to minus 0.4 volts, the dopamine orthoquinone is reduced at about minus 0.2 volts. So again, we have a change in current. And this is known as a cyclic voltammogram. So different substrates will have different characteristic voltammograms. This allows us to differentiate between these different substrates in the brain. So in terms of depicting this, now we can obtain 10 of these every second. What we've done here is basically to open up this waveform and now apply the potential in this direction. So if you're going from minus 0.4 to 1.3 back to minus 0.4, time is along the x-axis here. And in a 1 second window, we can basically stack 10 of these together side by side. And now what I'm showing you is current in the z plane. So the current is coming out of the screen towards you. And so what you can see here is a change in color, which is associated with that change in current. So we're using false color. And so this 0.6 volts here is where, again, dopamine is oxidized. What we can see here is a change in current that's associated with that dopamine oxidation. And if we use calibration techniques, we can actually convert this and say that over this 4 second window, we actually have an increase in dopamine concentration that occurs at this point right here. So it gives us, again, a way to look at these things in either an anesthetized or an awake animal while they're performing a specific task. And in this example, I'm actually showing you spontaneous dopamine release that occurs in the nucleus accumbens, the same brain region I mentioned before. But we can also stimulate this region. So if we stick an electrical probe directly into the dopamine cell body region and apply a burst-like stimulation pattern, we see a dramatic increase in the dopamine signal. So again, this false color, so the current is coming in the z-plane out towards you. You see a dramatic increase in that concentration. It's also shown here. So again, it gives us a good way to be able to measure these responses in real time while the animals are engaged in specific tasks. So now that we have all our tools in place, we can finally ask the question that we want to ask. And that is, does this Phasic dopamine activity actually drive drug-seeking behavior during cocaine withdrawal? So for this first experiment, what we decided to do was, again, to just apply a burst-like stimulation pattern directly to the ventral tegmental area. I've shown you that that leads to this increase in dopamine release. And we wanted to see what does that do to the behavior that I described to you earlier. So again, these are animals that have gone through cocaine self-administration, gone through 10 days of withdrawal, and then we're asking what happens when you stimulate this ventral tegmental area with this burst-like stimulation pattern. And what you see is an increase in their active lever responding. So again, they seem to be showing more cocaine seeking while we apply that burst stimulation. If you compare that to animals that had gone through saline training, so they never received cocaine, you apply the same behavioral paradigm, give them the VTA stimulation, there's no change in their behavioral responses whatsoever. So it's something about having experience of cocaine and having that pairing that seems to be important. So in this experiment, what we've shown is basically that inducing that burst-like stimulation is sufficient to drive the behavior. But the other question we wanted to ask, well, what if we prevent this firing from occurring? Does that influence the behavior? So now what we've done is to actually infuse a local anesthetic directly into the ventral tegmental area and to look at these responses. So this is in animals that receive saline infusion into the VTA. You see high active lever responding during withdrawal compared to the inactive lever. If we anesthetize the VTA, we lose that response. So the VTA, in this sense, is required or critical for this behavior to occur. So it's both sufficient and necessary. We also wanted to make sure that it was actually the VTA to nucleus accumbens pathway. So in this experiment, we're now infusing agents directly into nucleus accumbens, which block receptors that are specifically activated by that phasic dopamine release, which I described to you earlier. And if we do that, we again block the ability of these animals to perform this cue-induced cocaine-seeking behavior. So again, it gave us pretty good confidence that this phasic activity in the ventral tegmental area to nucleus accumbens pathway is actually critical for driving these behavioral responses. So if that's the case, the next question that we wanted to ask was, are there ways that we can actually decrease this response in terms of dopamine and decrease the behavior to try and decrease the cue-induced drug craving and relapse? So there had been some previous work that had looked at an area of the brain in the mesopontine known as the lateral dorsal tegmental nucleus. And basically what they did was to see whether this specific brain region actually influences burst firing activity of these dopamine neurons in the VTA. So what they did was to record from these neurons. Here they're showing the control situation where they have this irregular firing pattern that's associated with burst firing. But if they inactivated this LDTG, what they found is that these neurons transitioned again to that tonic or pacemaker-like firing. So they lost that burst firing activity. And they wanted to investigate why or what might have been contributing to that. So normally if you use the neurotransmitter glutamate in these neurons, you can actually induce this burst firing. So you apply glutamate, you see a dramatic increase in this burst firing. However, if they inactivated this LDTG, glutamate was no longer able to induce this burst firing. So others had previously shown that this area sends glutamatergic, GABAergic, and acetylcholinergic input into the VTA. Now the fact that you can no longer get burst firing even when glutamate was on board suggested that there was another neurotransmitter that was critical for allowing this to occur. So what we decided to do was to look at the acetylcholine aspect of this in particular. And now the basic question we want to know is whether this acetylcholine activity is actually regulating the burst firing of these neurons and the phasic dopamine release. So again, we're turning back to our voltammetry setup where we can actually electrically stimulate those dopamine neurons and look at the response. Except this time, we're also coupling this with a microinjection cannula directly to the VTA. So now we can ask what happens at baseline when we stimulate these neurons, and what happens if we have a cholinergic receptor antagonist, and then we stimulate these neurons. So just as a proof of concept showing you here, again, we're doing a burst-like stimulation of the VTA. At baseline, you see a nice increase in dopamine. If you infuse saline, there's not much of a change at all. So the first experiment we decided to do was to look at different types of cholinergic receptor drugs to see how they influence this system. So there are two classes of cholinergic receptors. There are the nicotinic receptors, which are ionotropic, so they can be activated, they can pass current through the channels themselves. There's also muscarinic receptors, which are coupled with G proteins. So we wanted to look at both classes of these and to use antagonists to block these receptors directly in the VTA. So when we do that, what we see is if we block the nicotinic receptors, we actually have a dramatic decrease in the phasic dopamine response. Similarly, if we block these muscarinic receptors, we have a dramatic decrease in the dopaminergic response. And I told you that glutamate is also important for this. These NMDA receptors are sensitive to glutamate. And so if we block the NMDA receptors, we again have a decrease in this dopaminergic response. So from a physiological standpoint, we've seen that these receptors are important. But we also want to ask, is this true from a behavioral standpoint? So I've told you that these types of firing responses and release are important for cue-induced drug-seeking behavior. I've shown you that these cholinergic receptors and NMDA receptors are also important. So our next question is, are these receptors important for the behavior itself? So the question we asked is exactly that. And what we did, again, is to take animals through our cocaine self-administration paradigm, through withdrawal, and then look at our cue-induced cocaine seeking. On the seeking test, we're infusing, again, agents directly into the VTA and then looking at the behavior over a period of time. So when we do that with a nicotinic receptor blockade, what we see is something that was very similar to what we saw when we inactivated the VTA. So if we block those receptors at this high concentration, we see that they're no longer performing this cue-induced cocaine-seeking behavior, so suggesting that these receptors are critically important. If we block the muscarinic receptors, we see a similar effect. Also, we saw an overall decrease on both the active and inactive level responses, so not as specific. Although surprisingly, with the NMDA receptor antagonists, we saw no change in their behavior. So it seems that these nicotinic receptors are critical for this behavior. These other two did not address or decrease the behavior as we thought they would. Now, if you think back to what I just showed you a couple slides earlier, I mentioned that all three of these agents actually decrease the phasic response. But then I also mentioned that this agent had no effect on the behavior. So there seemed to be a little bit of a mismatch there. But one thing I didn't mention is all of the voltammetry that I showed you was performed in naive animals. So the next thing that we needed to do was basically to ask, is there a difference in this phasic dopamine activity after animals have gone through cocaine self-administration and withdrawal. So we decided to perform that experiment, again, taking the animals through cocaine self-administration, a few days of withdrawal, and then performing the experiment with dopamine in the anesthetized animal after they had had this cocaine exposure and withdrawal. When we did that, we saw, again, a decrease with our nicotinic receptor drug, a decrease with our muscarinic receptor drug, but no change with this NMDA receptor antagonist. So it seems that something has changed in the animal's brain over time. That is, in a naive animal, you have a decrease that's occurring when you block these glutamate receptors. However, after cocaine experience and withdrawal, you have a shift where you no longer have that change, but you still get it with the mecamolamine. So again, highlighting these different changes that can occur in the brain with drug exposure. So a critical piece for us also, we've shown that these mechanisms are important for drug-seeking behavior, but we also wanted to see, is this specific to drug-seeking behavior, or is this important for general reward? From a therapeutic standpoint, what we're trying to do is decrease people's cravings for drugs of abuse. We're not trying to dampen the entire reward system. So if you remember before, I showed you that this system is important for both natural rewards and for drug rewards. So ideally, we'd be able to have a therapeutic agent that only targets the drug-seeking and not the natural reward-seeking. So to test that here, we basically asked, are these systems also mediating responses for cue-induced seeking for sucrose, for a natural reward? So the paradigm is basically exactly the same as what we've done with cocaine, except that these animals are now self-administering for a sucrose pellet. And we haven't done this in my lab, but there are labs that will use things like chocolate and banana. So obviously these animals are motivated for many of the sweet tastings that we're motivated for as well. And I'm sure several of you had some muffins this morning, so it wasn't an experiment. At least not that I know of.
We'll report on it later.
Okay. So basically in the animals here, we're taking them through 10 days of sucrose self-administration. Again, taking them through a period of withdrawal, and then now asking, will they press the lever for cues that were now associated with a sucrose instead of cues that were associated with cocaine? And they will. So we performed, again, the same experiment as we did before. So what we found here is actually if we use this nicotinic receptor blockade, we have no change in their cue-induced sucrose-seeking behavior. So this nicotinic effect seems to be specific to cocaine, not to sucrose. With a muscarinic antagonist, we actually do see a decrease for their sucrose-seeking behavior. So the muscarinic effect seems to be much more global. With the NMDA receptor blockade, similar to cocaine, we see no change in their behavior. So throughout, they're continuing to press the active lever much more than the inactive lever. But again, it highlights that this nicotinic piece seems to be specific to cocaine and not generalizable to rewards in general. So there's a lot of information on the slide, but I just wanted to give you a little bit of a schematic to show you the model that we're developing so far, based on all the data that we obtained. So what I'm showing you here, again, is the ventral tegmental area and the nucleus accumbens. And what we're proposing is that this exposure to these cues, discrete or contextual cues, can actually lead to an increase in acetylcholine in the VTA itself. That's sufficient to activate cholinergic receptors. I didn't have time to go through it today, but we've done a lot of work to try and isolate which subtypes of nicotinic receptors might be important. We have work to suggest that it's these specific nicotinic receptors that contain this alpha-6 and beta-2 subtype. This can activate a whole host of downstream mechanisms, which eventually likely increases phasic dopamine release, activates specific receptors, and induces this cocaine-seeking behavior. So just to give you a little bit more of what we're doing in terms of next steps, we're also trying to see if there are other targets within the VTA that might also be important. And one target that we came across are actually these L-type calcium channels. They're located within the VTA and have been shown to be important for some of this burst firing as well. So they've been shown to be important, particularly from transitioning these neurons to the from tonic activity to burst firing activity. And that's shown here. So based on this experiment, these investigators are using an L-type calcium channel activator. What they have is tonic firing these neurons here that shifts to this burst firing after this administration has occurred. And so we wanted to ask again, if this is important for burst firing, is it also important for the behavior that we're modeling? So we're performing the exact same experiment as before, except that now we're infusing L-type calcium channel antagonists directly into the VTA, asking what that does to the behavior. In the first experiment, we used an L-type calcium antagonist nifedipine, and again, we see a dramatic decrease in the active lever responses, so a decrease in the cue-induced cocaine-seeking behavior when this drug is infused. We also did another experiment using an FDA-approved drug known as isradipine and saw very similar effects. Now, this drug is actually used pretty heavily for cardiac-related disorders, And the advantage here is the fact that this is already FDA approved. There hasn't been as much work that's been done in the brain. But if there's some traction there, it may be easier to move this into the clinic in a faster fashion since it's already an FDA-approved agent. So something that we've been keeping in mind as well. Similarly, here we also wanted to make sure this was specific to drug seeking and not generalizable. And when we did that, we found, again, there's no change in the sucrose seeking. So if we infuse this rapine into the VTA, the sucrose seeking is intact. We also were curious, is this an effect that's specific to cue-induced cocaine craving, or does it also influence cocaine taking? So we did another experiment just to see, after the animals, after they've been trained to take cocaine, if we infuse this drug, does it change their cocaine taking behavior? And the answer is no. So again, this seems to be very specific to this cue-induced aspect that we've been looking at so far. And there is some evidence in the literature that there may actually be interaction between these nicotinic effects I talked about and these L-type calcium channel effects. So we wanted to do an experiment to see whether that is true for the behavior as well. So similar to our previous experiment with a nicotinic receptor antagonist, we see a nice decrease in the behavior if we infuse that antagonist into the VTA. In this experiment, we want to see whether there's an interaction between the two. So here I'm showing you what we found with the nicotinic receptor antagonist alone, a nice decrease. With the isradipine, the L-type calcium channel antagonist alone, we also have a nice decrease. If we add both drugs together, we don't potentiate that effect. So it's very similar whether you have one drug in isolation or both together. Again, suggesting they may be acting through similar pathways. So to get back to the schematic that I showed you before, now we've added another wrinkle to the pathway in a sense. That what we're predicting is that these nicotinic receptors normally can actually activate the L-type calcium channels, and that might also be important for regulating this response to the drug, or to the cues rather, through regulation of phasic dopamine signaling. So it gives us two targets that we can start to look at now. And obviously there are several other targets in this pathway. They may also be efficacious in terms of trying to think about potential therapeutic implications as well. So before I wrap up, I just wanted to mention briefly some of the work that we're doing now to try and look at the neural circuitry that's underlying these responses. So far I've focused particularly on these receptor complexes, but we're also curious about the circuits and pathways as well. So initially I mentioned this idea about this LETG that's projecting to the VTA that may be important. And what we're proposing is that it might actually be this LETG to VTA to nucleus accumbens pathway that's actually critical for driving these behavioral responses. So one way that we've gone about testing this is to start to use optogenetics, which some of you may be familiar with. And basically what optogenetics does is it's taking advantage of certain channels from bacteria that are sensitive to light. So what investigators have done is actually to harvest these channels and to be able to introduce them into mammalian systems. So in particular, this channel rhodopsin here is sensitive to blue wavelength light, about 470 nanometers. And when you shine blue light on these neurons, you actually can increase the activity of these neurons. Similarly, there's another channel known as halorhodopsin, which is actually related to chloride channels and can actually inactivate these neurons. So what investigators have done, you can basically take these receptors and package them in viruses to deliver to specific classes of neurons in the brain. And then it gives you a way of specifically activating or inactivating those neurons. So all the work I showed you up to this point was using electrical stimulation. So basically we're taking current and passing it into a specific region of the brain, but we're activating everything that's in the general vicinity of our electrical probe. The advantage with optogenetics is now that if you have neurons that are infected with these classes of either channel rhodopsin or halorhodopsin, and you shine the light, you're only going to activate or inhibit those neurons that express the channel of interest. So it gives you much more specificity. So what we've done initially is to try and use this methodology to try and isolate this pathway. So again, we're curious about the LETG projections to the VTA. Now, if we use electrical stimulation and activated the LETG, we would be also activating all the neurons in that region and everywhere they project to. But what we're trying to do is to isolate only those neurons that project to the VTA. So what we've done is actually to take our channel rhodopsin, to package it in a virus, and infuse it directly into the LETG, wait for 4 to 6 weeks, and because we have a green fluorescent protein tag, we can actually look and see whether those neurons got infected. So I'm showing you neurons here in the LETG, in the cell body, and their terminals in the VTA. So as a next step, what we did was to take our optical probe, take a specific wavelength of light, and actually shine it directly into the VTA. So now we're only isolating neurons in the VTA that contain this channel rhodopsin, and the only neurons that do are the ones that projected from the LETG. The next piece was then to try and actually see whether this influences this phasic dopamine response in the nucleus accumbens. And what we found is that is indeed the case. So if we activate only those neurons that project from LEG to VTA and record the nucleus accumbens, you can see this nice increase in phasic dopamine activity that I mentioned earlier is important for this drug-seeking behavior. So what we're doing as the next step is to see whether the same manipulation can actually enhance this cue-induced cocaine-seeking behavior, or if we inactivate these neurons, does it decrease the response? And again, giving us much more of a grasp on circuit-level responses that are important for some of these behaviors. So before I wrap up and open it up to questions, just want to talk a little bit about some of the clinical implications of this work. So I put this in the context of cue-induced craving and relapse. And what we found so far is that these nicotinic receptors and these L-type calcium channels are able to robustly decrease cue-mediated behaviors if we block these channels themselves. And we've shown that particularly with cocaine, but we have some evidence both from our lab and others that suggests that this might be a more generalizable phenomenon for other drugs of abuse as well. So actually, a colleague at Yale, Jane Taylor, has done some previous work looking again at nicotinic receptors in the VTA and showing that they also seem to be important for what's known as conditioning reinforcement. And that's basically the availability or the ability of cues to drive responses. And she saw this for ethanol-associated cues. So again, these nicotinic pieces seem to be important now for cocaine and also for ethanol cues. Again, I've shown you our work here looking at cocaine-seeking behavior. There's also some work in literature looking at whether you enhance the activity of acetylcholine in the VTA, and that seems to be able to increase cue-induced heroin seeking. So now we have phenomena that seem to be similar between different classes of drugs of abuse, and again, that gives us some traction in terms of trying to think about potential therapeutic implications. And importantly, I've told you that what we found so far is nicotinic and L-type drugs don't alter cue-induced sucrose seeking, so it's not just a general dampening of the reward system, but seems to be specific to drugs of abuse. We've also done other work, again, looking at cue condition reinforcement, and these nicotinic receptors don't seem to play a role in food-associated cue behaviors as well. So giving us more confidence, again, about the specificity. And again, highlighting that there may be some therapeutic advantage to trying to look at these as potential targets to treat some of these disorders. So in this last slide, basically the point that I want to make here is that we're just doing, this is a small sliver of work in terms of the bigger scheme of things and the work that's being done in the field. The work I've been showing you today is actually focusing on this aspect here, the VTA to nucleus accumbens pathway and how it's important for craving. But of course, there are other pathways important as well. There's been a lot of work that's focused on the prefrontal cortex in terms of the role that it plays in terms of inhibitory control. I think someone actually mentioned that in an example when we were going through earlier. But that can also influence how people respond to cues in the environment. But we're hoping that our work, even though it's a small sliver here, is actually making an important contribution. And we're excited about some of the work that we have going forward as well. So my most important slide before I finish is, of course, thanking all the people who performed this work. It's been a great team to work with over the years, and we're continuing to move forward. A lot of the work that I showed you today was actually performed by a former postdoc, Wojtek Selecki, who's actually a faculty member in Poland at the moment, finished up his postdoc and moved to Poland. He worked with a talented group of individuals, some undergraduates, Shay Behrens and Blake Swerling, who helped him out on that project. Robert Wickham is a graduate student in the lab who contributed as well. It's actually pictured here. He's getting ready to defend his thesis in the next couple of months. And also have some other folks who've been jumping in and helping out with the L-type calcium channel project, specifically Eric Nunez and Shannon Hughley, and a host of other undergraduates and graduate students who've helped out over the years. So if we have time, I guess I'll open up to questions. Thanks so much.
So we have about 10 minutes for questions. So if you want to, you can raise your hand and we can try and get you a microphone just so that the video camera can pick up what you're asking.
How is it that when the colored light is used to activate neurons, that the light is inside the skull to activate different brain areas? So how is that procedure done?
That's a great question. So he's asking how the colored light is actually inside the skull when we do the optogenetics. So basically, in a way that's similar to what we did with the behavioral pharmacology, so we have a cannula that we can actually insert into the brain to target specific regions. So we can use that cannula and actually so it's basically like a very small, needle isn't the right word because it's not pointed, but it's basically a small track that allows us to deliver drugs. We can use something similar to actually deliver light. Basically, so if you've ever, so if you go online and just search for optogenetics, there's actually videos online. And so the fiber is called a optical fiber. It's actually connected to a tube that then goes all the way back to the laser. But what you'll see is that blue flashing light that's occurring in the cage, and basically that's going all the way down to the specific brain region that you're looking at. And people have done a lot of experiments to try and see how much spread there is of the light in the brain tissue, and it seems to be pretty confined in terms of where it's going. But yeah. It does look like a flashlight, like a blue flashlight that's occurring in the page. So it's a good question.
So I was just wondering how you would specifically target the nicotinic receptors in the VTA in a therapeutic setting?
Right. That's a great question. So in all likelihood, we wouldn't specifically target the VTA nicotinic receptors. She's asking how we would target those nicotinic receptors in the VTA from a therapeutic standpoint. There has been some previous work that was done using systemic administration or oral administration in humans of this mecamylamine, and they did see changes in cue-induced craving in cocaine-dependent individuals, but there were also a lot of side effects that were associated with that. So what we're proposing here is that the VTA may actually be critically important for that effect, but since there were side effects associated with the mecamylamine, we would actually propose to use more selective targets. So again, now that we see that these specific subtypes of the nicotinic receptors seem to be important, that may be a better, it might be better to use a drug that targets those specific classes rather than using mecamylamine. But you're right, we wouldn't target just the VTA, but we would hope that systemically or orally administering would be sufficient to influence those receptors to decrease the behavior. Great question.
Thank you, Dr. Addy, for a wonderful presentation. That was fantastic. And I also want to just mention, too, that was one of the best explanations of cyclic voltammetry that I've ever heard. So I really appreciate that. Thank you so much. My question is simple. It's just related to your behavioral model that you showed in the beginning of the talk. I was really surprised by the fact that when you did the cocaine cue pairing with the animals, they really didn't seem to extinguish even after 45 days. Could you just, I guess, mention why you think that might happen, and if you lowered the dose, would they extinguish, or if you did a different drug, would it be more pronounced?
Right.
Which is
so
very
that was actually
surprising.
Between subject design, so we're actually using different. But you're just curious why they still were showing the robust behavior that far out. So people have also done. It's funny because the question I usually get is why the behavior didn't increase. So people have actually done some experiments looking at what's known as the incubation of craving. So sometimes after longer withdrawal periods, you actually see even a greater response. So if you compare 1 day to 30 days to 45 days out to 6 months, you actually see that response increase over time. So people have different hypotheses about different changes that are occurring in the brain that may actually be contributing to that. For your specific question, I'd propose that the plasticity in the brain or the changes in the brain that are associated with the cocaine and the exposure have not diminished at 45 days, and that's why we're not seeing any extinction or extinguishing of the behavior. If you went far enough and some of those things had started to change and decrease, then I think you would start to see that drop off. I think that's probably what's maintaining the behavior. Good question, though.
Yeah. Hi. Thank you so much for the wonderful talk. And actually, a new question came to mind after hearing this question. It reminds me a little bit of gambling, right? So the reward is so small, statistically, but gamblers keep pressing the lever, right? So it seems that the ventral tegmental area is definitely involved in gambling addiction to not necessarily just reward, right? So my question was, when you at some point anesthetized the ventral tegmental area to show that it's necessary and sufficient for this behavior, did you stimulate the ventral tegmental area itself while it was anesthetized, or did you also stimulate other brain areas while the ventral tegmental area was anesthetized?
We haven't done that yet. But yeah, that would be another way to even address it.
Right, because it would reveal other areas that could be responsible for this.
Exactly.
Yeah.
Exactly. Yeah, and you could get some compensation as well. That's a great idea. Oh, question here?
So I was wondering, you mentioned circadian rhythms earlier, and I was wondering if the phasic period, like the phasic period of the neurons firing, and the what was the other part of the, what was the other like type of neuron fire?
The tonic versus the burst or phasic firing?
Yeah, tonic versus phasic. Would that be different, like during like sleep cycles, like the phasic firing with the tonic firing?
That's a great question. I actually don't know the answer to that. So most of the work that people have done at sleep cycles is looking at other regions of the brain. I have to go back and see if people have looked at that. I would still expect that you would have both tonic and phasic firing happening, but I don't know if there could be a shift in different sleep cycles, but I actually don't know off the top of my head.
And the reason why, the reason why I asked that is because like it seems to me like through withdrawal and relapse, it seems to be like a rigid pattern, and it just seems like a cycle. Like you could lose sleep over like it would affect your sleeping patterns, and then it just affects like everything, and just makes you want to keep like it makes like the person who's addicted to the drug, it just makes them cause them to be in a rigid cycle where they just keep like they go through a withdrawal period, they want to keep doing the drug. It could affect their sleep cycle with their circadian rhythms, and then
People have definitely looked at some of those interactions. But I think your question is great, but yeah, I don't know off the top of my head. I have to go back and see if anyone's looked at that.
Thanks.
Great question. This microphone coming.
The channel data was really, really exciting and very interesting. That it clearly separates between nicotinic and muscarinic. Now, it seems like our endogenous system, like feeding, is primarily muscarinic.
Seems that way, yeah.
Which means that would it be too premature to assume that all the exogenous acquired drugs, like I'm saying, like cannabis, amphetamine, could line up in that pathway as well?
That's our prediction. Yeah, that's our prediction, exactly. But yeah, it's been interesting to see every manipulation we've done with muscarinic seems to be more important in terms of even just feeding behavior in and of itself, whereas the nicotinic seems to come online after exposure to drugs of abuse.
Then the body must have somehow organized it. Right, this is exogenous and this is endogenous.
Right. But we're still
Without
trying to
knowing.
figure out how,
Yeah.
how that occurs. Great comment.
We have time for this last question here.
So just another question about the behavioral paradigm. If you look at repeated exposure to the cue alone trials, how long does it take for that behavior to extinguish?
Right, so typically when people do an extinction paradigm like that, they'll go anywhere from 8 to 10 days before that does actually extinguish. And they'll use a criterion to say that the animal shouldn't press at any point within 20 minutes. So once the animal stops pressing for 20 minutes, then the behavior is considered extinguished. Good question.
So with that, we're at quarter after. Our workshops begin at 25 after. So what I'd like to do is just thank Dr. Addy again for showing up and for a fantastic talk.